great. How are you feeling? Good. Well, I'm glad to hear it. Yeah, no, I am loving my semester. If you want to know the truth, I have great classes. I'm like on top of things. I'm not behind on anything, which feels nice. I'd like to keep it that way. I have a student teacher, and she's wonderful, which is always a little nerve wracking because you're going to get a student teacher and they'll be like not wonderful.
She's really nice.
Yeah, she's really nice, and she's already like excellent. She's doing great work. So my job is to just get out of the way. So yeah, I'm doing great. Life's pretty good. Solid C plus. So
What would make it?
sure, yeah,
Oh, it's
pretty good grade, right?
no. My mom went around for 20 years.
Yeah, I mean, again, I come from a different era where grades were just assigned differently and meant different things. So I don't think in the same terms. Like I think the courses I liked and was okay at, I would think like that'd be like a 70, right? But you guys, obviously, grades have just been handled differently with you guys, which is fine. Like that's not some big difference. Anyway, we'll focus. So, lesson 2.6, and this is going to be called dihybrid crosses. And really, we're just going to do one example together to kind of get the idea across. And then hopefully from here you see that like we can look at as many traits as we want. So if you remember when we talked about a monohybrid cross, what did we say those words meant? One trait. So in this case, we're looking at, yeah, that's right. So this is just tracking two different traits. How's that? Wonderful. Tracking two different traits. I'm going to say when crossing two organisms. And again, remind me when we say crossing two organisms, what do we mean? Yeah, breeding, right? Reproducing. So we're going to stick with Mendel's pea plants because whatever, that's sort of traditionally where we start. Yeah, it's good. Let me know it's my turn, and then I'll take my turn back. So let's just dive right into it. Example, pea plants. And we're actually going to stick with flower color, which we just looked at, and we're going to look at seed color. And we're just going to remind ourselves of the alleles for each. So we said that for flower color, we had big P, and that related to what color? Purple. And we had little p, and we said that that was white. Good. And the fact that I have drawn the p as a capital and the little p means that purple we are saying is dominant, and white flowers we are saying are the recessive trait. Good. What about seed color? Does anyone remember we looked at that example the other day? Yeah, you can buy yellow peas at the store and you can buy green peas at the store. Green peas are more common to buy, but actually yellow seeds are the dominant trait. So you can have yellow seeds or you can have green seeds. Okay. So now we're going to have to keep track of two traits together. So a plant that is purebred for purple flowers but heterozygous for seed color is crossed with a plant that is heterozygous for flower color, and has green seeds. All right, I want to give you a minute to write that down. All right, take your time. And then what I want you to do after you've written that down is I just want you to tell me the genotypes of the parents. So take a minute and work out the genotypes of the parents. I'm just going to color code these parents here because I think sometimes, the problem is no matter what color I highlight it, it's going to relate to like a color that's in this question. So you're just going to have to live with that. Actually, I guess blue is safe. What else is safe? Yeah, okay, that's safe. So I'm gonna get rid of this yellow because that feels like it's begging to be confusing. And you know me, never confusing. That hurt my feelings, but that's okay. So take a minute and work that out, all right? Sorry? Yeah, and we haven't done this before, but that's okay. I want to see what you come up with, and then we'll talk about it. So we want to talk about the first parent, and we want to talk about the second parent. Okay, the first parent. Tell me about the genotype for flower color. Okay. Does that make sense? We said they're purebred, which means the two alleles are the same. What's another word for purebred? Homozygous. And in this case, is that the dominant or the recessive? Dominant. So we'd say homozygous dominant. And then what would we say about their seed color? They're heterozygous, which, close, but for seed color. Yeah, that's fine. And so we just write them right next to each other, because it's got both cases, right? It has two copies of the color of the flower color and two copies of the seed color. Hello. And by convention, we just write them next to each other. Does that make sense? If you're looking at 3 genes, you just write the next two next to that. If you're looking at 5 genes, we'd write them all in a row. See if that'll help you, if you haven't already, write out the genotype of the other parent. You just write them right next to each other like that. That's the convention. And again, kind of the formatting is we've got 4 slots we need to think about, right? Which is we need to know what are the alleles for flower color. You don't need to write this if you don't want to, but I just kind of want to make sure we see it there. And these are the alleles for seed color. And again, why do we have to have 2 of each? Nope. On the right track, because they're diploid. They have 2 copies of each, right? Okay, so what would we write for this parent that is heterozygous for flower color and has green seeds? And then small y, right? Does that make sense? And again, these 2 are the 2 copies that tell us about flower color. And these 2 tell us about seed color. Cool. How's that feel? Not too bad? Feels all right. Okay, so now let's just jump right into the Punnett square. But the first thing we need to note is what are the possible gametes, right? Well, what is kind of the general form of a gamete? Well, it's not diploid, it's haploid. But at the same time, we're going to be talking about two alleles. Not two copies of the same allele, but it's going to have an allele for its flower color. And it's going to have an allele for its seed color. Does that make sense? Is that logical enough? Right? How many possibilities are there? And I shouldn't really write these far away from each other because we always write them just like that. So seed color. So what are the possible combinations? Well, you could be dominant for flower color and dominant for seed color. Or you could be dominant and recessive. Or you could be recessive and dominant. Or you could be recessive and recessive, right? Does that make sense? We won't necessarily see all these. We might. But those are the only possibilities, period, right? When we're looking at two genes. So now let's actually dig into it. But the point being, how many ways can you combine these? We just saw 4 ways. So when we do the Punnett square, can we put just 2 things on the top anymore? We need 4, right? And likewise on the side, we need 1, 2, 3, 4, and going up and down, we need 1, 2, 3, and 4. Okay. Now maybe, like me, you didn't leave yourself really any space up here. If you did that, I'd just encourage you to just highlight everything and just move it down your page a little bit. I know, right? I'm very helpful. And maybe you made it a little bigger than you want it, in which case I would just encourage you to highlight it and shrink it a little bit. I feel like mine could be a little bit smaller, really. And then maybe you'll find that you just missed a line altogether. That's okay. We can just come in and fill it in. Maybe you failed miserably in making a Punnett square is what I'm saying. Probably you haven't, but Lord did I. But that's okay. Because, okay, ready? Do I have the technology? Can I do this? I think I can. Ready? Ready? Yeah, there we go. There we go. And then you know what? While I'm at it. And then, you know what? I want them all to, like, kind of line up. What? Stop judging me. You don't see me judging you, do you? Yeah. Okay, I do. That's true. But only because I care. Also because I don't like you. But those are the only 2 reasons. Because I care and because I don't like you. You'll be all right. You're tough. Okay, so there's my Punnett square. And again, why do I need 4 slots? Because if we have 4 alleles altogether, can be combined. Maybe we call this x1, x2, y1, y2. These can be combined 4 ways. Whatever your alleles are, you could have x1 paired off with y1, right? You could have x2 paired off with y1. You could have x1 paired off with y2, or you could have x2 paired off with y2. There's 4 possible combinations to make gametes from the allele. So first we talk about, what was this one? Was it big P, and then hybrid? Yeah? Take a minute and work out what the gametes could be. And if you're feeling confident about that, then do the same here. I'm sorry. Oh, I lied. That's not what that is. Those were both lowercase, right? That's too messy. Let's just try again, Joe. So yeah, work out what the possible gametes are. And if it helps to kind of do the same way I teach sort of binomial distribution in math, if it helps to kind of think, well, those two could go together, or those two could go together, or those two could go together, or those two could go together, if that kind of helps you work it out, then great. If not, it's tricky. No, it's confusing. That's fair, right? What this basically means, you can think of this almost as a cell starting off, and it's got, a DNA chromosome here that's a big P, and it's got another chromosome floating around that has a little p on it. And it's got another chromosome floating around that's a little y, and another one that's little y. And the question is, when we undergo meiosis, what are the possible ways you could put together those chromosomes? Because we know these all have 2 copies, but these ones only have 1 of each, right? So it means that this one here could end up with this one here. That's a possibility, right? But it could also be the case that this one here, that purple one, could end up with this other one down here, right? Or we could have this little one here ending up with the blue one, right? We could have that little p. And so you just have to kind of work out all the combinations, all the ways they could split up. And if we assume they're splitting randomly, it's basically the first one compare with the first one, or the first one compare with the second allele here, or the second with the first and the second with the second. 4 combinations. So for our friend up here, what are the combos? This big P allele could end up with that big Y, in which case we'd get what? Big P, big Y. All right, cool. But the big P could also end up with the little y, in which case we'd get, why am I putting that down there? Big P, big Y. Or we could get the big P paired up with the small y. And then, even though this ends up being the same, it's a different combination. This big P could end up with the big Y, which would give us big P, big Y. Or this one could end up with that one, which would give us big P and small y. Does that make sense? Those are the combos. Yeah. It is 1 for each. It is 1. That's a great question. Very good question. And let's talk about why this is okay. It is 1. It is 1 flower color gene in each section, right? And it is 1 seed color gene for each section. So when you do a dihybrid cross, how much are you going to have in each section? Well, you're going to have 2 alleles, but they're going to be talking about 2 totally different things. Does that make sense? Each gamete will have 1 allele for flower color, and it will have 1 allele for seed color. Did that answer your question? That was a great question, by the way. Does that make sense? What you're not going to do is break this into 8 pieces and say, well, they could pass the big P on, or the big P, or the little p, or the little p, or the big Y, or you know what I mean? Like, we're not going to break it down that way because a gamete, a sex cell, is going to have 1 of each, right? So what we're figuring out are what are the ways those can combine. So again, here we have flower color, and here we have seed, right? So in each case, we have just 1 copy of each gene. That was a very good question, by the way. Thank you for it. Okay, down here, let's go in order. The big P could end up with the little y, or the big P could end up with the other little y. So we get 2 of the same. Does that make sense? What's the other possible combination? The little p could end up with the first little y, or that little p could end up with the second little y. Does that make sense? We have some repeats here, but that's okay. So these are the possible sex cells, also known as what? That's right, of the second parent. Looking at these 2 traits, there's 4 possible ways that parent can make sex cells. As it happens, 2 of them are the same and 2 of them are the same. So really, we get 2 possible outcomes, but they are different. Because in one case, we're getting the little y from its mother. In the other case, we're getting the little y from its father. Yeah? Sorry? Yeah, they are haploid. Very good. So gametes are the name of sex cells. Haploid is an adjective. So gamete's a noun it means sex cell. Haploid's an adjective, which means a cell that only has one copy of each allele. But absolutely, you're completely right. These gametes are haploid, whereas the parent cell was diploid, which means it has two copies of each. Yeah, definitely. Good question as well. Nice. All right, now when they come together, we're going to make normal somatic cells. In other words, everything that goes in here is a new pea plant. But a new pea plant has how many copies of each allele? Of each allele. 2 for a total of 4. 2 for seed color, 2 for flower color. Does that make sense? So when we make this, if this seed comes together with this pollen, what would we get? Well, we get big P and big P. Can we see that? And we get big Y and little y. Does that make sense? Cool. What about here? What'll we get? Will we? Careful.
Oh, no, sorry.
Yeah, yours may be the same because you may have gone a different order, and that's okay. Big P with big P and little y with little y. Cool. What about here? Yeah, same as the big one, right? Big P, big Y, little y. What about here? Big P, small y, small y. Now, the nice thing is, since this is the same, what will we know about this row? It'll be the exact same. Can we see that? This row is the same as the previous one, so we don't actually have to work it all out. We can just copy our row above. Is that going too fast for anyone? Does anyone not understand why it's okay to just copy that row again? Be honest, because my experience has always been that if one person's confused and says it, usually there's actually like 8 people who are confused and they're all just being shy. I know some of you can be very shy. Others, less so. Okay.
Fun now.
Uh,
Like, not the order, but like how we got
to here?
yeah, like this part, like the top.
Sure. Okay, guys, can we refresh the top? Okay, everyone put your pencils down. Pencils down. Pencils down. Has everyone promised the pencils are down? Let's do just a different parent together. Let's make a new parent, same two. Let's make new traits. Let's say we have an allele that can be either X or little x, another one that can be G or little g. Okay, I'm just making stuff up. We don't care. Okay, and let's say we have a parent who is big X, big G, little g. Does that make sense? Okay. This is a parent cell, which means if you wanted to, you could think of it as being like, there's the cell, there's the nucleus, these are the chromosomes. And one of those chromosomes has a big X on it, right? One allele. Another one has a little x. This one has a big G, that one has a little g. Great. When it goes to make sex cells, how many different ways can it combine those? Well, okay, it makes a first sex cell, and maybe in this case, this X chromosome ends up moving over here, and so does this G. In which case, what would we have here? Big X paired with big G. Does that make sense? But there's another way they can make a sex cell. That same big X might end up paired with little g. What will this look like then? Big X, small g. Is one of those more likely than the other? No, that's just as likely as that. Of course, this leads to the other combos, because if the big X ended up with the big G, then what did the little x end up with? Yeah, then the little x ended up with the small g. Does that make sense? On the other hand, if the big X ended up with the small g, well, then what did the small g end up with, or the small x end up with? Does that make sense? So those are the only, and if I'm not missing, there's no other ways these can come together, right? Does that make sense? So if I was putting this into a Punnett square, rather than drawing cells, I would say, okay, from this parent, oh, that went badly. From this parent, what are the possible sex cells we could make? Well, I'd say if I have 2 alleles we're looking at, there's 4 possible, or 2 genes we're looking at, there's 4 possible combinations. Big X can go with big G, or big X can go with small g. Little x can go with big G, or little x can go with small g. Does that make sense? The order doesn't matter one bit. Does that make sense? So if I had chosen to do this by going, if I'd started by saying little x can end with little g, that's fine. And then little x can be with big G, and then big X can be with big G, and big X can be with little g. That's not going to matter one bit. That's the exact same answer. Did that answer your question?
that order matter?
Okay, that was a good question. By convention, when we put them together, just be consistent. So if you put X's before G's, then always put X's before G's. Does that make sense? If you put G's before X's, that's fine. Just do that. And then the other thing is, no matter which parent they come from, if there's an uppercase and a lowercase, by convention, we put the uppercase first, then the lowercase second. So we'd write this, not that, regardless of where they got each one. Does that make sense? So the rules would be whatever order you pick to put them in, doesn't matter, but be consistent with it. Don't put flower color first the first time, but then the second time now you're putting seed color first, then the third time you're back to flower color first. That'll get confusing. So we try to be consistent. And then if they're the same, it doesn't matter. But if it's heterozygous, we always write heterozygous as dominant, then recessive. It doesn't matter how they lined up on the chart. Did that answer your question? Cool. Any other questions? That was a really good one. All right, then somebody tell me what combo is going to go here. What am I going to write? So do you want to try this one? So armed with our knowledge. So if this and this come together here, what do we get? Yep. Very good. You want to keep going? Awesome. Good. Then here? Yeah, big P, little p, big Y, little y. And then y's are the same. Good. And then the nice thing is, again, if these two are the same, do I need to really think my way through this? No, I can just copy this down here, right? Big P, little y, big Y, little y, big P, little y, little y, big P, big Y, little y, and big P, pair of little y's. Whew, lots going on there. Two traits. One trait, we needed just a 4 square, right? 2 by 2, 4 square. Two traits, how many squares do we need? How many squares is this?
16.
16. How many squares do you think you need for 3 traits? I don't know, maybe we'll find out later. So now we get to the misery part, though I don't think it's that miserable, which is, let's look at the genotypic frequencies. Okay. Now, what are the possible genotypes we could be looking for? Well, we know we could be homozygous dominant for flower color, homozygous dominant for seed color, right? Okay, what are other possibilities? We could be heterozygous and homozygous dominant, right? We could be homozygous recessive and homozygous dominant, right? Does that make sense? Can we keep going? What are other possibilities? I'm not saying from this. I'm just saying overall possibilities, right? Then we could be dominant here and heterozygous, heterozygous and heterozygous. We could be recessive and heterozygous, right? And then finally, we could be dominant with recessive here, heterozygous, that should be a P, with recessive, and recessive with recessive. Those are all the possible combinations, right? Can you see that? Is that a lot? I feel like it's a lot. So now let's count. How many of these do we actually see in our chart? Do we have any like this? Do you see any in there? No. So that would be 0 out of a possible. How many squares do we have?
16.
And what percentage is that? 0%. Okay, so we have no plants that will end up being homozygous dominant for both traits. What about heterozygous for flower color but dominant for that? No. So 0 of 16, which is what percent? 0. Okay, so far so easy. Bless you. What about this here? Nope. The reason all those have to be 0 is because we have no way to have a homozygous dominant for flower color. Do we see why? If we look at our parents, this parent has to pass on what allele? The recessive seed color allele, right? Okay, now looking back at our chart, do we have any of these? Yep. Yeah. Let's count how many. I'm going to color this in this lovely green color here. How many look like that? Well, that guy there, right? And that guy there, and that guy there, and that guy there. Anywhere else? No. So how many is that?
4.
4 out of, which is what percentage? 25. Cool. Okay. Do we have any of these combos in there?
Yep.
Yeah, let's start finding them. There's one, and below it's got to be the same, and there, and there. So how many is that? 4 out of 16 in this particular case, which is 25%. Cool. Okay, let's keep her going. Let's go with this little orange one here. Do we have any of these guys? None?
Oh.
Nope, we don't, right? So we can skip that. What percentage is that? That's 0 out of 16, which is 0%, right? Okay. Do we have this next one?
Yep.
Yeah, that orange is really aggressive. I'm going to go with a slightly less aggressive orange.
Yeah.
Kind of lovely. All right, how many?
4 of them.
Yeah, we have them there, and there, and there, and there. Okay, so how many is that? 4 out of 6. Oh, that's a highlighter. Fun fact. 4 out of 16, and what percentage is that? 25%. Awesome. And do we have any of...
Yes.
Do we have any of these guys? And that is our remaining 4, right? And that's 4 of 16. And what's that as a percentage? 25, right? And then finally, this will be 0 out of 16, and that will be 0%. Okay, whew. That was a tricky one. Yep, what's up? Is this a chemistry question? Give me a few minutes, okay? Okay, and then obviously if we've talked about genotypic frequencies, what's the last thing I'd ask about? What's phenotype mean again?
The look of it.
Yeah, what's genotype mean again?
The actual chemical makeup.
Yeah, in other words, which what? Which alleles it has, right? And for every trait you look at, how many alleles will each one have? 2, right? So if you're looking at 2 traits, you're going to have 4 alleles to think about. If you're looking at 3 traits, you'd have to think about 6 alleles, right? And if you were looking at 4 traits, you'd have to think about 8 alleles. I do not mind zooming it in. Sorry, I'm trying to get everything in one spot. Is it here you're looking for? Or the table itself? Yeah. So now, let's talk...
Wait, I'm confused. I was still running something else.
Gentlemen, one sec. We done? We good? Okay, good. Thank you. Yeah, carry on.
When we were writing down step C,
Yep.
like the possible phenotypes,
Yeah.
but those aren't.
They're not all possible from this combination, right?
So did we just do that?
And by the way, if I just did this by imagining every possible combination, if I was reading it from the table, I would have probably not bothered with all these. Does that make sense? But this is not from the 2 plants we bred, but this is a possible genotype for 2 pea plants. Does that make sense? Now, how many of those did we get? None. If I didn't tell you that you had to list every possibility, I would just stick with the ones you actually saw. Does that answer your question? So yeah, I wouldn't do all this work every time. Maybe I should have done it this time, but it did. Now again, can you imagine that if you were trying to think about 3 traits, does this get pretty complicated? 4, 5, 6? What about the estimated, depending on how you count, 100,000 to 300,000 genes that we have? And the thing you'll find is that no computer on Earth could possibly handle dealing with calculating monohybrid crosses with all those traits. In other words, if you took the most powerful supercomputer on Earth and plugged into it all of your mom's alleles that she has and all of your dad's alleles that he has, you can get a genome taken now. You can get your DNA tested, and they can tell you what copy of every single gene you have in the human genome. It costs about $10,000 right now, I think. A lot of money, but you can do it. But even if you had all that information, they couldn't then sit down and map out all the possible combinations. No computer can handle that yet. It's a massive amount of information. because this grows in complexity exponentially, right? Every time you look at a new trait, you're multiplying by 4 how many things you got to look at, right? All these combinations, yeah. Sorry? Oh, good question. Alleles are those different versions of a gene. So if the gene tells a plant what color its flowers are going to be, that might be like if you were to go look in the plant and maybe it has a chromosome sitting in there and it turns out that right here is the gene that controls flower color. And then if you went in and zoomed in on it, that might be 80,000 base pairs. You know what I mean? If you zoomed in on that, what you'd find is that it is like A, G, C, C, G, C, T, A, G, C, blah, blah, blah. And of course, what would pair up with that? What letter would be here? T, C, G, G, C, G, A, T. Is that G? We're going to call that a G. So we're going to then say this has to be a C, and that's a G. Okay, so that could be that like gene right there. And maybe this is the allele for purple. And then, you know, a white, often, when things can either be colored or white, often all that white pigment is actually just that the pigment's broken. Does that make sense? So it could just be as simple as there's a mutation at some point where that pigment that's supposed to color purple got broken. There's a gene mutation. And so it could honestly be the case that the allele for the white flower is just A, G, C, but then right here, this codon got changed for some reason and just got filled in with T's. Does that make sense? And now, so if we look, this section of it here changed. Can you kind of see that? And we can make, so that was AGC, so then we said that there, kind of changed. What would pair up with this, just so we can do it? T, C, G, A, T, C, G, dot, 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 right? So this could just be a second version of the gene. This is the broken, right? I.e., in this case, it's just white flowers, allele of the same gene. In other words, if you want to find this gene in the chromosome, you'd go to the same spot on that chromosome for every pea plant out in the wild, and then you would look at that section of DNA and you'd say, do they have this version? Okay, they have the purple allele. Do they have that version? Oh, they have the white allele. Does that make sense? There's some genes that have 30, 40, 50 different alleles, right? We've just looked at cases so far where there's 2. When we talk about blood typing, you'll see that your main blood typing has 3 different alleles, which is why you can have A-type blood, B-type blood, or O-type blood. And then there's a whole second gene that has 2 alleles, which is why you can be Rh positive or Rh negative. Does that make sense? But there are a lot of different alleles out there. And oftentimes, if the allele is a little bit different but doesn't change the trait, when we're looking at the animal as like a big thing, we say, oh, that's the same allele. You know what I mean? Even though there's slight differences, if it didn't change the color, we'd say, oh, that's the same allele. But if you're a molecular geneticist who's going down and looking how related are these things, well, then you might look and say, oh, these 2 alleles are different by 3 base pairs. Even if it didn't change the trait, you'd say the fact that those alleles are a little bit different tells me that they can't be like direct offspring from each other likely because there's been some mutation there. And it's the more mutations there are that differ them, the less likely they are to be related. What theory of species relatedness does that refer to? The one where we're looking at the DNA to see how similar and how different they are? Anyone remember? We said when we look at this just shape and form, we call that the what concept? Morphological concept. Good. If we're looking at sort of how their biology functions, where these might, this is a bad example, but if all we did was change this C to a G and maybe the gene doesn't work any differently, then in the second concept, we'd say, oh, they work the same. They're the same. That's the biological concept. But here, where we're actually comparing base pairs and saying how similar and different they are, that's what concept? The phylogenetic concept. Does that make sense? Hopefully, learning about how your DNA works actually makes a bit more sense of that. Because now we can look at different animals on Earth and actually break down right to their DNA. And like we talked about that alcohol dehydrogenase gene, we have that in our bodies. Kyla Caron, who's at Brock and comes in for help every once in a while, just did a lab at Brock where they had to take fruit flies and break them apart and analyze their alcohol dehydrogenase gene using something called gel electrophoresis, which we will likely do this semester. But that's a gene that fruit fly shares with us. But their allele for it's a little different than ours. Anyway, I hope that answered your question and didn't just make it more confusing. Can I ask, like, as a percentage, what percentage of the time I answer a question do I make it less confusing? And what percentage of the time do I make it worse, would you say?
90 to 10.
I can live with that. That's pretty good.
Which one is the worst?
Well, I'm just assuming it's good. Don't hurt my feeling here, Cora.
Leave them in suspense.
No, I'm good. So now the last thing is, we've talked about the genotypic frequencies, but of course we need to look at the phenotypic frequencies, right? And in this case, let's just talk phenotype here. We don't have to go through all the possible combinations. This combo here, what would their phenotype be? What would their flowers look like? Purple. And the seeds? Yellow. Okay, cool. What about this fella here? Purple and yellow. And this fella here? Purple and green. And purple and green. So all together, we only have two phenotypes here. The first is purple flowers
100%.
and yellow seeds. And those would be what percentage? Well, we have to add these together, right? 4 of the 16 were this one, 4 of the 16 for that one, for a total of 8 of 16, which is 50%. Good. And what's the other phenotype we saw?
Purple flowers and yellow green seeds, sorry.
You don't need to be sorry. Would you be sorry? For misspeaking? Oh my God, if I had to carry on every time I misspoke, we'd never get anything done. Kind of like now. How many of these? Yeah, 8 of 16, which is 50%. Okay. Dihybrid crosses are clumsier, but the place, most people do great with this stuff. When people screw it up, where they screw it up is always just getting these filled in. Does that make sense? So I thought really quickly, let's just practice not doing the whole cross, all right? But you'll practice doing the whole cross tonight. But let's just practice filling in the gamete spots. So I want to do Punnett square, but I'm not going to care about the actual square right now. I just want to practice filling in those gamete spots because it's always the place people make mistakes. Does that make sense? So just need a 4x4 grid. And I'm just going to pick two totally random letter combinations. Let's say here we have a parent who is big Q, big M, little m, okay? And somebody give me a genotype for the other parent.
Little Q. Nope.
Little Q. Little Q?
Yeah.
Okay, and then for M?
Big M.
Big M, little m again?
Yeah.
Awesome. So I just want you to fill these in. You don't have to do the whole thing. We can imagine this gene could be whatever you want. Q could stand for quaint. And little q would be not quaint. What's dominant trait? Quaint or not quaint? Okay, don't. Okay, so Paul and Orlando, I'm just going to have a good old-fashioned nervous breakdown.
That's so real.
Oh yeah. Just get a little bit of like menthol for under the eyelids. You do this the good old-fashioned way. Just think about your childhood puppy. That's what I did. Oh. So again, you don't have to do the whole Punnett square. I just want to make sure we can figure out what the gametes would be, right? So if this is the parent who has 2 copies of each allele, what do we call that again? Yeah, dominant, recessive, but we say the diploid. And these need to be the possible gametes, right? And gametes, of course, are are haploid, right? So remember, if we're talking about 2 traits, then the parent has to have 4 combos, 2 for each trait, right? Whereas if we're haploid, they just have 1 of each, right? Yeah. Yep. Yeah, that's right. That's right. So this here would be the genotype of a diploid organism, where it's going to have 2 alleles for each gene. And this is the genotype of a haploid cell, which is just going to have 1 of each. That was a great question, by the way. Very good. You guys ask good questions. The order doesn't matter, but what matters is consistency. So there's just some manners to it. So the first rule is, if I have established that we're writing Q first and we're writing M second, which means we're always going to write Q first and M second. Does that make sense? There's nothing magical about that, but it's like driving on the right side of the road. Nothing special about driving on the right side of the road. You can get by driving on the left side of the road just as well. They do it in England. They do it in Japan? Question marks. Do it in Singapore. But if you go to do it here in Canada, what's the problem? You're going to hit someone because everyone else is driving on the right. Does that make sense? So once you've established that we're going Q then M, then that's what we're going to do consistently. And then the other piece of manners is, if you have an uppercase and a lowercase of the same thing, we always do the uppercase first, then the lowercase second. Does that make sense? And other than that, no. But if you're making the question up yourself, you can do whatever order you want. And in terms of how you pair them up. So what are the possible combos here? And again, I'll show you how I kind of think my way through it. This big Q, what could it end up with? Well, it can't end up with another copy of Q, because then this would have 2 copies of Q, but if it has 2 copies of the same allele, then it wouldn't be haploid, it would be diploid, but gametes are always haploid. So what can the big Q end up with? Big M. Because remember, in meiosis, during anaphase 1, a copy of each chromosome goes to each side. So yeah, we get big Q paired up with big M. What's the next possible combination? What else could big Q end up with? It could end up with the little m, right? And then we're going to get some repeats here, and sometimes you get some repeats. So the other thing that big Q could end up with is the other big Q could end up with big M. Now, that's the same genotype as this one. It got it by a different way. This is mom's big Q with mom's big M. This is dad's big Q with mom's big M. But that's okay. It still works. And then what's the last possible combo? Big M, small m? Is that allowed? No, because those are the same gene. Can you have 2 copies of the same gene in a gamete? No, that'd be being diploid, but gametes are haploid. So that big Q can be with that little m, which again is the same as this, but it got there a different way. What are the combinations here? And again, just draw arrows. This little Q, what can it end up with? Big M. Or it can end up with small m. Okay, and then we move on to the other Q. What can this end up with? Either big M or small m. And that's it. Does that make sense? And now if you wanted to, could you fill the square out? Sure, I'm not going to do them all, but just for the sake of argument, what would this one here be? Big Q, little q, big M, big M. Okay, and you could just keep going all the way through. Does that make sense? And again, don't forget that when you get rows that are the same, you don't have to start from first principles. I'm going to post a little bit of homework for this up on the Teams page. Just a little bit of practice. I'll throw it in OneNote. This stuff is straightforward, but it takes some practice. So take your time and practice it. Yes? Yeah, I've decided to give it to you tomorrow instead. It's not a big one.